welcome to the first night of See and Learn 2023 that we have a breeze. It's still been hot all day, but we finally have a breeze. So thank you for coming, everyone. We have a special evening tonight, so we're going to get a double header. So let me get started. So first of all, to our live streaming audience, thank you for watching at home, and thanks to Carib Trans, who supports our live streaming efforts. Tonight's speaker is kind of co-sponsored between Hummingbird Villa, one of our beautiful homes on the island, and the traditional cottage of Hidden Garden, which is truly beautiful inside and out. Our presentations, as well as our entire program and foundation, are not possible without the support of Prince Bernhard Culture Funds, as well as public entity SEBA, which is our government, and also the many, many sponsors who make this event possible. We have grown from early beginnings of only 5 sponsors to more than 10 times that. So we have at least more than 50 sponsors who sponsor this event in many ways. So if you recognize some logos, next time you're in their locations, please give them a wave and a thank you. Then we have, new this year, part of our 20-year anniversary, we did the soft launch of the Isle Explore platform. So this is something you can take a picture with your phone for the QR code and start using it on the island. If you're new to the island, you'll see there's many nonprofit cultural organizations that you might not have known of. You can also use the iNaturalist app, so you can identify creatures, plants while you're out hiking. All kinds of interesting interviews with local people. So it's a really cool platform, and they would like your feedback. So in a month or more, they're going to even take it to another level. We have raffle tickets. Have you heard? But for our live streamers at home, you can contact us via phone or email if you'd like to purchase. We will make it happen. They're only $5 and over $10,000 worth of prizes. As you can see on there, there's everything from handcrafted items to ferry tickets, sunset cruises, week stay in St. Croix, and many wonderful villas, including Larry and Sherry's flamboyant cottage, who are in the audience this evening. So please be sure to support that, and the money goes to a good cause. Us. Helping us make these events happen. Upcoming events. So tomorrow afternoon, there is still space on our main speaker's field project. So this is a marine project, so you do need to be a diver, and she's going to tell you a little bit more this evening of what she's going to do, but it's super interesting and fun. Our next location is going to be very special as well. So on Thursday night, we're going up to the rainforest to the Ecology, and if Dixie can stand up and show herself. So she is our cacao expert, so that means chocolate. So the Ecology promises a special menu that is chocolate inspired, and it will be fun, and there's a mini shark presentation before that talk. And then Friday afternoon, we're going to hike up to show you a cacao tree, explain more about that. And then it's planned that you can go to Chez Booba afterwards, where there will be chocolate-inspired martinis. So sign up at the tent, and we'll be taking care of you there. If you participate in any field project, we have complimentary T-shirts, tank tops that you get for that, as Mia is wearing tonight. Right behind us, if you're a live streamer, maybe you didn't know about this, but as part of our 20th anniversary, a scientist, Erica Moulton, supported us by having a beer created, and it's a great beer. Who can raise one in the audience? So feel on top of the world with a Saba cloud top that is inspired by Saba Spice. So we have a little mini, and we do mean mini. It's going to be a very short presentation, but we think it's very valid. And I think the best way to introduce her is to just tell you a little bit about what happened yesterday. So she is the traveling companion of Dixie, our cacao expert, but she specializes in backyard poultry So people who have chickens and raise eggs. So yesterday we had a little get together with the people on Saba who raise eggs, and she really raised their awareness and taught them more about how to increase their egg production, biosecurity, and risk of disease. So we're going to introduce you for our short-term mini speaker to Miss Katie Ockert out of Michigan State University. So here she is.
Good evening. Thank you so much for having Dixie and I here. This has been a lovely experience for both of us, and everybody has been so welcoming, and we'd just like to thank you all for that. So, like Lynn said, I am with Michigan State University Extension, and I have kind of a dual role. I help people learn how to manage livestock and also manage wildlife. And then also with that, how we can prevent diseases and keep our animal populations, both domestic and wild, healthy. So some of the things that I'm going to share with you really do relate to that larger ecosystem that we have on this planet. So there's this concept called One Health. And today we were down by the harbor, and there was a mural that said something about Saba symbiosis or symbiotic. Has everybody seen that? Yeah. One Health is exactly that. So our world is all interconnected. We as humans, our health and well-being depends on the health and well-being of our animal populations, both domestic and wild. And both of animals and people health depends on the environment's health. When we have factors that have been going on, such as population growth and expansion into new areas where we're encroaching on wild animals' habitats, and we're deforestation, population growth, that just opens the door for all sorts of diseases and bacteria and things that we then get exposed to, and then we travel around and carry them in the environment. Because if you think about how mobile people are, I mean, where is everybody from? We're all from different corners of the world here tonight, right? We travel so easily. And sometimes some of those things that we don't want to travel with us, travel with us. And this morning I was kind of looking about biosecurity in terms of marine life, because being on an island, that is really important. Introduction of invasive species is a really challenging topic, right? It affects your wildlife on the island. It affects your waters. It affects the ecosystems in both places. So this One Health concept has been adopted worldwide by the World Health Organization, National Institute of Health, Centers for Disease Control, all of those things. And it's just a really important concept to think about anytime that we talk about the health of animals and people as they relate to one another. So what is biosecurity? Very simply, biosecurity is the steps that we take to prevent disease spread or pathogen spread. If you recall, during COVID, everything that we did as humans to slow the spread of COVID was biosecurity. Washing our hands constantly, creating distance, regularly cleaning and disinfecting things. Those are important steps and practices to maintain in everyday life, whether it's for your own personal health, or if you have animals, or if you're traveling around the world in a boat, for example. I read about ballast water. Okay, so we're from Michigan. We have the Great Lakes, but like we're in the middle of Michigan, so we don't really interact with water, so I don't know a lot about marine stuff. I'm a land girl. But there's a lot of different opportunities for cross-contamination in our environment just by virtue of living. Our shoes, like if we blacklit our shoes, you'd probably be really disgusted. And we're depositing those things all over the environment. So always things to think about as you travel out and about, and especially if you are in agriculture and you're going from farm to farm, it's always really important to practice some of these biosecurity steps and principles so that you don't take diseases from one farm to another. So there's a couple different kinds of ways that pathogen can be transmitted. So the first is vector-borne. So if you think of Zika, West Nile, those kinds of mosquito-borne or bug-borne viruses and bacteria. Then there's airborne and waterborne. So COVID. water or airborne, right? Proximity or direct contact with animals. So that's wildlife or domesticated animals. And then foodborne illness. And there are just a lot of different safety precautions that you can take, especially when it comes to direct contact with animals, and then also foodborne illnesses. So always cooking your food to temperature is really important. I know I like a really rare steak, but sometimes that's not a good idea depending on where you're at. But one of the things that we talked about yesterday was washing eggs. Do you wash your eggs? Do you not? It depends on what the producer does. So if the producer washes your eggs, then you gotta put them in the refrigerator because the eggs have pores in them and that lets bacteria come in. So it's always just important that before you use your eggs, whether they sit out on the counter or whether they're in the refrigerator, that you just give them a good wash before you put them in your pan or however you're gonna cook them. And you also wash your hands. So just things like that really help. We talked a lot about different poultry diseases. Salmonella is the number one that affects probably every single one of us. Has anybody had salmonella poisoning? Oh my God. Oh yeah. Bad, huh? Yeah, it's no joke. So salmonella is one of those really prevalent diseases that chickens carry. It can be transmitted both foodborne. That's why it's super important to wash your eggs, wash your hands, any surfaces that you touch while you're preparing any poultry products, whether that's meat or eggs, and always cooking those products to temperature. And then direct and indirect contact. So if you, I notice there's lots of island chickens, right? So anytime you're in proximity of their manure, or if you do have chickens and you're holding them, they can harbor that bacteria on their feathers, on their feet, on their beaks, all over their bodies. And if you don't wash your hands and practice some good biosecurity, you are at risk for getting salmonella. So those are just a couple, that's the main poultry-borne disease. But of course, worldwide we have diseases like high path avian influenza that has decimated flocks in the United States, millions and millions of birds. So you probably all experienced an egg shortage earlier in the year due to high path avian influenza. And that's a disease that migratory birds carry. So it can happen anywhere, anytime. So it's always something that protecting your animals is a good idea. Limiting contact between wild birds and domesticated birds. There's some really key biosecurity practices to remember as you interact with all birds. So in closing, and this was super short, so I hope you learned something, simple practices make a huge difference in both animal and human health. Just washing your hands, cleaning your shoes, making sure that you're conscious about the environment, what you're leaving in the environment, always will help keep people and our wildlife healthy. So thank you so much. Yeah, questions? Does anybody have any questions? All right, thank you.
Thank you. Thank you, Katie. Okay, here we go. So our keynote speaker tonight is Lauren Simonitis. Have any of you ever seen an animal ink underwater? Yay. What animal was it? Squid? Okay. Squid? Okay. Octopus? Cool. Cuttlefish. Laura Simonitis, cephalopods. Cephalopods include squid, octopus, and cuttlefish, and some other lesser-known species. They are well known to ink, but our presenter tonight will talk about other less-known inkers. Lauren Simonitis is an aquatic sensory biologist interested in how animals use their sensory systems to understand their environment and impact their role in predator-prey relationships. From the prey perspective, she focuses on how animals use ink as a chemically mediated anti-predator defense. Lauren also studies the olfactory system of a common ocean predator, sharks. By describing the general morphology, distribution of sensory structures, and fluid dynamics of different chondrichthyan, also known as cartilaginous fish, nasal morphologies. Give a round of applause for the Ink-redible Lauren.
Thank you so much. That was awesome, and thank you so much for that presentation to start. That was an amazing opening act. So I am so happy to be here talking to you guys today. Like Erin said, I don't think we will ever give presentations in such a beautiful place. Normally we are like stuffed in a conference room, so this is quite the upgrade. And thank you so much to the See and Learn Foundation for bringing us out and for having this amazing event. I'm going to talk to you guys about ink, and I'm going to talk to you about shark noses, and those may seem like two very different things, but they are actually pretty related, or I've made them pretty related because they're what I want to study, so that's how they've been related. I am currently at Florida Atlantic University in South Florida, but I also work at University of Washington, which is not Washington, D.C., the state. It's the state, so it's like furthest in the continental United States you can get, so it's not a great commute, but it's awesome. And I'm really excited to chat with you guys today, not just because of what I study, but because my family's from the Caribbean. I'm really happy to be talking about science in the Caribbean. I love chances to talk about my science to areas where I'm from and where my people are from. So my mom is from Cuba, and like every photo I have of my family in Cuba, they're always on the water, they're always fishing, and the water is just such an important part of our lives. So I'm really happy to be able to be somewhere that's so close to my roots and be able to talk to you guys about what I study and who I am as a person, as well as what I'm interested in. But my whole journey into this realm started with ink. And like Lynn said, you guys might think of cephalopods, so squid and octopus, but this is Simone, and she is a pygmy sperm whale. So something that is not super well known is that there are two species of whales that ink. And you can see her here. She is in a rehab situation, so she's stranded. And she was brought into Moat Marine Lab in Florida, and they were trying to rehab her and relocate her to the ocean. And whenever pygmy sperm whales are really stressed, they do something that is pretty common, which is to release waste. So if you've ever grabbed a frog or grabbed an animal and it gets scared, or a dog expresses its anal glands, that's something that we see a lot across animals. But inking animals have like added a little something to it to make it extra nasty to and say like, get off of me, please let me go. And she's really interesting as well because what you can see is just the absolute incredible volume of ink that she produces. So, and then she kind of goes and hides in this ink. So what is ink even to begin with? Because it is kind of a hard thing to define. But we have kind of these three main cast of characters that ink, and they have some things in common. So first off, we have the cephalopods, squids, octopus, cuttlefish. Then we have sea hares, so that's the second panel right there. That is Aplysia dactylodema. So they're mostly the Aplysia species. I'm going to refer to them as Aplysia for the rest of this presentation. They're very cute, and they're my personal favorite. They're called sea hares because they have little bunny ears, which are their rhinophores up there at the top. And then lastly, we have those pygmy and dwarf sperm whales, those weirdo whales that I like so much. But trying to go into each of these categories a little bit more, we have cephalopods. So here what you're going to see is a common cuttlefish inking. And something really important that they do is they change colors as they ink. So a lot of us on yesterday, I forget what day it is, but yesterday we saw an octopus on the reef, and as it moved across the reef, it changed colors. And we see this happening in tandem with inking behavior as well. So they try to match their body color to the ink, which helps with the predator defense. And we'll talk about that a little bit more later. Finding Nemo also makes this very easy for me because a lot of people are familiar with this little cute octopus Pearl. They have melanin-based ink. So melanin is what gives our hair color, our skin color, our irises color, and that is what's giving the color of this ink. It's normally this dark black, sometimes purpley ink, and they also mix it with mucus. And mucus is really cool because they are actually able to, they're so smart, they are able to figure out how much mucus they need to display that they want. So what we can see at the top is we see these like little lines. Those are called ink ropes, and that means that there's a lot of mucus, so it's being nice and pulled out long. Then we have what's called a pseudomorph up here. A pseudomorph is basically a little decoy squid, so it's like, look, this is my little squid, but then I jet away and leave you with this ink squid. And then what we see is when there's not really that much mucus, we can create an ink cloud. An ink cloud. But it's gone. And now it's back. So the next guys are the sea hares or the aplysia. They are my favorite. They create this purpley, sometimes red looking ink. And again, they kind of have this variation in what they can do. So they're adorable, and they have, if you look when the video comes back up, there'll be like these big tendrils, but there's also this ink cloud. And just to kind of put into perspective, these were the first ever animal I worked with as a freshman in the University of Miami when I was first getting started in marine biology. So we've been together for a very long time, and they're very near and dear to my heart. But their ink is based with a red algae. So they eat red algae, and they take that red algal pigment, so what gives the algae color, and that is part of their ink. So that's what gives it that red purpley color. What we see is that whenever you feed sea slugs like lettuce or a green algae, they'll still create ink because it still works as a defense. It's just no longer red. So the only thing that's coming from their diet is the color, but the actual like nasty chemicals in the ink are made within their own bodies. And then they also have a mucus, which is what we call opaline. And opaline is this, like, it comes from a separate gland, and they can do the same thing where they vary how much ink and how much opaline they produce. Opaline is like this mucusy, globby stuff that whenever it comes in contact with water, it goes through this chemical reaction and polymerizes. And yeah. Yeah, it's cool. It's a very visual presentation. There you go. So what you can see here is on the left and the right. These are two different secretions. So the one on the right-hand side, the purpley guy, that is a lot of ink. And then you can see the amount of bubble, and like it's hard to see mucus in a picture, but it's really mucusy. And then that's more of an opaline-driven response. And this is my amazing undergraduate intern, Anahi, who is now doing her master's at Cal State LA. And she is looking at basically if we keep bothering them, they will keep changing how much ink and how much opaline makes the correct mixture to make us go away. So they're pretty smart too, and they can kind of control these two little secretions together to give us this perfect like ink response. And then finally, again, we have the pygmy and dwarf sperm whales. So what you see at the bottom here is their feces, because that is how I get their ink. So they actually ink within their digestive system. But I'm just going to give a quick introduction to these guys a little bit more, because I think that these are kind of our more weirdos. So I call them the weirdo whales. They're weird in a lot of ways. They're in the family Cogidae. There's pygmy sperm whales, and there's dwarf sperm whales. And you can tell them apart kind of easily. A lot of times I used to work in marine mammal strandings, and we would get a call, and it would never be the correct one. But they kind of look a little bit different. So pygmies are a little bit larger than the dwarfs, and from the surface they have a little bit of differences as well. Pygmies are rounder, and they have this little like stubby dorsal fin, versus our dwarves have flatter backs, more of a typical dolphin shape, and then more of a typical dolphin dorsal fin. And these guys are weird because they have a lot of defense mechanisms. So the first is what we call Batesian mimicry. And this is when you want to look like something that is dangerous in order to not get eaten. And these guys particularly look like sharks. So again, whenever I was on a stranding call for them, we always knew it was a pygmy or dwarf sperm whale because they got reported as stranded sharks. And part of that reason is they have that really blunt head. They have this subterminal mouth like a lot of sharks have. And they even have a false gill slit. So if you can look at this picture of a stranded one, he has this little white line right here. So that was often referred to as a false gill slit. And they have really nice counter shading. So their bellies are white and their backs are gray. So they're really trying to pass as sharks. And again, they have that weird predator defense of inking. So for a while, we thought that they were just eating a bunch of squid, because they do eat a lot of squid, and then they were pooping out a bunch of squid, and that squid happened to be full of ink. But then at Moat Marine Lab, a mother-calf pair stranded, and they brought them in, and the calf was still only drinking milk from mom, so she was not yet weaned, but she was still producing milk. I mean, she was still producing ink. She was only drinking milk, and she was producing ink. So my lab, we actually went through their digestive system, and we do something called histology, which is where we slice super thin. Same thing that happens if you ever get like a sample taken at the hospital and they send it for a biopsy. We can do that on a lot of different tissues as well. And when we looked inside of their digestive system, we found melanocytes. So these are areas that create melanin, and there were these little pockets of melanocytes all throughout their digestive system. So they create the melanin on the walls of their digestive system and then shoot the ink into their colon. And then whenever they are scared, they let out all the ink. But what's also in their colon is poop. So it is a nasty mixture of poop and ink. So I would get these jars of poop in the mail, and my whole lab would be so mad because then I have to go through them and it smells really bad. But I'm probably the only person that gets super happy when I get poop in the mail. So it's awesome. And I have a bunch of partners all throughout the United States who send me poop. So it's a really nice relationship that I have. And these guys are super understudied, so we don't know much about cogids in general. Like, they're very solitary. They don't swim in pods, so they're usually just alone or like in mother-calf pairs. They're kind of small for whales, so they're not like very easy to spot. All that we know is from strandings or from like opportunistic like observations out at sea by like fishermen or by research vessels. So they're really understudied, and their ink is even more understudied. So the work that we're doing is some of the first ever published work on this ink. So we kind of have an idea of who uses it, but now I want to talk about how it works. And our main use is as an anti-predatory defense. And to talk about predation, we need to first understand what happens during a predation event. So first we have the initial encounter, and I want you guys to think about what senses are being used, because I am a sensory biologist, so we're going to talk about the senses. 5 senses plus a little extra because we're in the water and we got some fancy ones. But the first thing is the initial encounter. So this is where the predator and prey are both observing each other using their long-range senses. So vision if the water is clear, but sense of smell is like a really good one underwater. Hearing is pretty good as well. Then we have the attack. So this is when the predator is moving in on the prey, trying to take control of the prey item. Next, we have the capture. So in this example, it is when the seal is within the shark's mouth. So it's whenever the predator has control of the prey item. It could be whenever they grab it if they have hands. And finally, we have ingestion. So this is when the prey item is moving down the predator's digestive tract. Now, from the prey perspective, we have some different threat levels here. So we have general versus acute threats, and the acute threats are really mostly on the latter half of that. So we do have animals that can actually defend themselves in this latter half. So animals that are poisonous are attacking in the ingestion route. Animals that have like helmets or neck teeth or spikes, they're stopping at that attack whenever they're making contact. But then when we think about the general ones, that's when they can start with those long-range senses, which are vision and olfactions, or the sense of smell. So ink is an anti-predatory defense, and it works in 3 ways. It works visually, so like you guys saw in those videos, it's a very dramatic visual response. It is this big smoke screen Houdini situation. It also works chemically, so there are chemicals in the ink that smell bad. And it works physically. So because it has that mucus, it's really goopy, it gets on everything. A lot of times, like something we use all the time is lobsters. We'll spray ink at them, and they spend a bunch of time cleaning themselves, trying to get the goop off, instead of trying to eat the prey item. And ink is used a couple different ways as well, if we think about what the techniques are for this. And the first is phagomimicry, and this is a fancy word that means like decoy, right? So if we have a squid in this example, the first thing that the squid is going to do is it's going to use those long-range senses, and it's going to see or smell the shark coming. And then it's going to change into this dark black coloration and release that pseudomorph, so that little fake squid. So from the shark's perspective, it saw a squid, it turned black, now there's still a little black dot, and it's going after that black dot. The squid is then going to change to transparent and swim away. But the shark still sees that little black squid blob, so he's going to go and get a mouthful of ink. Where the chemicals come in is it's thought that maybe those chemicals in the ink smell good to be like, ooh, come eat me, I smell delicious. So it's both a visual and a chemical response in theory. Then we have alarm cues. So this is a source of community care. So if your buddy is inking, that's a great sense that something's wrong, so you should probably leave. And these sea slugs, they're not super mobile, but they do swim. And one of the best ways to get them to swim is to expose them to the ink of a conspecific or an animal of the same species. So if you pipette on ink from a aplysia onto another aplysia, you'll get them to start swimming. It's really cute, and it's really like angel-like. I think they're adorable. And then we have defensive chemicals, and kind of the best example of this is a skunk. So this is a chemical that smells bad. If something smells bad, you don't want to eat it. But it can go the next level, and it can be aversive, so it can get in your eyes and your nose. It can burn. It can sting your orifices. So we kind of have these different ways of using ink as an attack mechanism. And one of the things that I think is just fascinating about studying ink is thinking about convergent evolution. And what convergent evolution is when unrelated organisms use something similar to deal with a similar threat without being related. So this is not something that was passed down through evolutionary history. These animals are not inking all because they have some inking ancestor that passed down an inking structure, an inking strategy. All these animals evolved the same response to a problem despite not having any interaction in evolutionary history. So a great example of convergent evolution is echolocation. So bats and dolphins use the same technique of sending sound waves back and getting that response to see what's going on when they can't visually see, and using sound to replace that sense. And so for my work, I really focus on these three guys, and I compare them in this like comparative convergent evolution sense. So my main question when I was thinking about this was, these guys are super unrelated. So do they have a similar effect on a common predator? So despite these guys learning very differently how to attack, if we tested all of these inks on one common predator, would it have the same response? And I chose the very terrifying bonnethead shark. And if you have ever met a bonnethead, they are maybe 3 feet long on a good day if they like stand up super straight. They are not scary. And they have this stupid little mouth and this stupid little head. They're not even a good hammerhead, because they didn't get the memo on the hammer. They have a bonnet. A little shovel. So, but these guys are really nice and easy because they are really easy to catch. Hammerheads are notoriously very difficult to keep in captivity or to not get super stressed out. If you look at them weird, they just give up and die. But these guys are pretty hardy. They were pretty easy for us to catch in the local waters. And in that nice hammer, they have a giant nose. So it was really nice for us to be able to test a chemical system on a big chemical sensor. And also what's great about these guys is a lot of the work of the colleagues that I work with, they've shown that no matter what shark you test, so we have tested the sensitivity of a handful of shark noses that all look really different. We've tested hammerheads with these giant organs, normal sharks with these like pointy organs. We've tested their relatives, the skates and the rays. And despite what their noses look like, they all have the same sensitivity to smells. And also if you're like a big shark fan and you think they're really scary and cool, they actually are not any better at smelling than any other fish in the ocean. They have the same smelling abilities. So sorry to burst any bubbles. But what's cool about that is what I learn on these little cute, easy to manage guys, I can apply to some animals that might be a little bit more ecologically relevant that are actually eating these whales, or actually targeting them. But these guys do eat squid, so it's not like they've never encountered an ink attack in their life. So I had these sharks and they were just free swimming in a tank, and I decided to bother them every day and see how they responded. So I used ink from the Aplysia, from the cuttlefish, and the Kogia, which is the pygmy sperm whale. And I also use some controls. So whenever we do behavioral experiments, we want to make sure that we have, if we're using a negative control, something that we think is going to be negative to them, we want something to show that they either can be not bothered or have a positive response. So we use seawater just to make sure that getting something shot in your face wasn't bothering them. We use food coloring to control for the color. And we use food odor to show something that is positive, a yummy response. Food odor also really helped because we could test if the shark's nose was still working. So if you guys have ever gone to a bakery and it smells so good as soon as you get in, but then by the end of the time that you're checking out, you can't really smell that cookie anymore. That same thing happens across noses. So we wanted to control for that. So if we kept hitting them with this food odor and they started behaving inappropriately where they were either avoiding it or they just weren't interested, we knew that the shark was done for the day and that any responses that we got were not going to be valid anymore. We had this camera filming overhead, and we looked at locomotory kinematic variables. And basically what that means is we looked at how the shark swims. So we looked at different things like speed or angular velocity or all of these different metrics. We're going to go through one today. But what's cool about this is if you look, this is me painting my shark. So these are little white dots. And if you've ever watched like behind the scenes of like how they film like CGI movies or how they make video games, you see them wear those really silly ping pong ball suits. And that's what I was doing to my shark is I was giving him a little ping pong ball suit because what I can do is I can take this footage and put it into very similar programs and I can see this little stick figure shark and see how it's swimming in the tank and how it's responding to these outside forces. So this is as if you are my camera and you're standing overhead of my tank, and then this is my shark swimming freely. So over here, we have these two little white lines that are the tubes that go outside of the tank, and my amazing interns were crouched underneath the tank so the shark couldn't see. And we have these discs that sit at the bottom of the tank, and then these little straws that come up about 4 inches off the bottom. So whenever something was injected, it was injected into the water column, not along the bottom. And then here is my shark, and I was standing on a platform overhead getting ready to tell one of them to shoot whenever the shark got close enough. Okay, good. So whenever the shark was like about a body length away, we started ejecting ink or whatever was what we were testing, and then the shark came in contact with this ink and responded in some sort of way. So those kinematic metrics I mentioned before is speed, angular velocity. There's a lot of cool ones, but this is kind of my favorite one, so we're going to talk through this one today. So this is angle of deviation, and basically what that means is how much did the shark deviate from its initial swimming path. So here is the response to seawater, and here the shark is in its initial path. So it's swimming kind of by this injection site. That's that disc that's sitting at the bottom. And we see this little initial bubbling happening here. And then we see the response to seawater, which is only really a 20-degree variation because sharks do swim, you know, pretty sinusoidally, pretty snaky, pretty slithery. So that's pretty normal. That's like a non-response, really. We see the really big opposite response to cuttlefish. So here again is the initial bubbling, and here is that blue disc, and the shark is approaching it, and then bam, we see over 180 degree turn away from the ink. If you look at that bottom corner, you can see that big black ink cloud that results. They hated it. They did not like it, which was great for us, but not so great for them. And then what we can see here is if we look overall at our results, this was really awesome for behavioral results because if anybody has ever tried to look at the behavior of an animal, even like with your dog, it doesn't always listen to you and it doesn't always behave the same way every day. So this was 4 years of footage. And my interns were amazing and went through so much of it with me. So I just have to give everybody who worked on this props. But this is a lot of work distilled into one graph, which I think is like a great great thing about science is we can do like your entire life's work in a graph with like 5 bars. But here we have like the response to each. So the red is Aplysia, the purple is cuttlefish, and the K is Kogia. And if the shark moved away, it was given a negative value. If the shark moved towards it, it was given a positive value. So overall, we saw really negative responses. And these were all significantly different than our controls. So food coloring and seawater, the sharks didn't care. They just swam on by. They were really unbothered. And then we see the opposite when we look at food odor. We saw an overwhelmingly positive response. So when we look at these videos, we can see the shark turning away, and you'll kind of see this play as I speak about this. But all of our inks reacted the same way. They all elicited this big avoidance response. Here you can see the response to food odor, where the shark comes in contact with that blood and then turns around and just bites the ever-loving crap out of that thing. I had to replace it so many times. But yeah, it was a great response. Something that was interesting is that our food odor was eliciting an attraction response, which was opposite of what would happen if that ink was acting as like a chemical decoy of like, hey, come eat me, I smell good. So in this instance, we do not think that ink is working chemically phagomimetic. And what we see in other people that test this is that phagomimicry, that like ink blob decoy thing, is really only a visual response. It does not have to do with the chemicals. Whenever they get in contact with the chemicals, they do not like it. And this makes a lot of sense if we think back to Simone, because Simone loved to hide in her ink. And if your ink smells good, it doesn't make a lot of sense to hide in like a sundae if you're trying not to get eaten. She's just really cute. I love her. Okay, so I kind of had this like parallel done. But kind of like what my next steps are and what I'm currently working on at Florida Atlantic University, and I do a lot of this work down in the Florida Keys, in Keys Marine Lab, is looking at, are they actually smelling the ink? Because what we have is these really cool responses, but we don't really know what is going on inside of their nose. Because it is possible that they could be swimming through with their mouth open and they could be tasting the ink, and that tastes bad. So we really want to make sure that the nose is what is being attacked here. So we have this, my mom calls this the Frankenstein setup. She does not like when I send her pictures of this, but this is a shark that we put into tonic immobility. So we turn him on his stomach and they go kind of asleep. Do not do this in the wild with sharks. Please do not do this. People like to watch videos on Nat Geo of people flipping sharks and them becoming little puppies for belly rubs. This is not something that should be done if you are not trained on this. But we flip them over and we put these electrodes into their nose. And the electrodes are basically like whenever you get hooked up to an EKG and the electrical signals of your heart come up on the screen. We can see the same thing in the nose, because whenever the nose smells something, it sends an electrical signal to the brain that's like, hey, I'm smelling something. So we can pick up on that electrical signal and see if the nose is actually responding to something. And we just started collecting this data down in the Keys. This data was actually I started this project during my dissertation, and I started March of 2020 was my first data point. And then I was shut down for a year and a half. So I pivoted, and now it's my postdoc. So now this is what I'm working on. And this is us getting our first ever ink responses. This is like so many years of work, again, in one photo. But it was an amazing day. And I just want to show you guys again kind of how we introduce that ink. So this is a dye trial. So here you see the shark, and it's being ventilated with that tube in its mouth that keeps fresh water flowing over its gills. And I do want to say again that all this is permitted by our ethics committee. We do go through extensive ethical reviews to make sure that we're treating these sharks as well as possible by finding out this information that is critical to their survival. So what's going to do, it's going to zoom in on their nose, and you're going to see us putting dye through it to make sure that the shark is actually, this stuff is going through its nose. So that's that dye going into the one side, out the other. And we'll talk a little bit more about how a shark nose works in a minute. Because now I kind of have this response. So I see that sharks are smelling ink and that they are not liking the smell of ink. So this is a really nice predator-prey interaction. And probably like a lot of you, I thought that sharks are so famous for their noses. Like they are swimming noses, and you know they can smell a drop of blood in the Olympic-sized swimming pool, and they go crazy. And then when I looked into the literature, that's really not true. We don't really know anything about shark noses. So I had a lot of information about how this attack worked, but I didn't have a lot of information about how the sensor works. So how are they sensing this? So now that is a really big focus of my research program, is looking inside the head of a shark. And I do this by doing CAT scans on a bunch of sharks. So we soak them in this dye that's just like if you've ever gotten when you have to drink that nasty liquid before you get an MRI or a CAT scan done. We do the same thing to them. So that's what allows us to see their soft tissues, because normally x-rays only pick up on hard tissues. Sharks don't even have bones. They're very, very squishy guys. They do not show up well in x-rays. So we soak them in this dye, which lets us get these beautiful images of what's going on inside their head. And I'm going to kind of show you what a shark's nose looks like, because it's not just this nice little nostril here, it's this beautiful network that lives underneath the skin. So here's that same shark, but not in x-ray. And if we flip him upside down, I am going to show you a photo of a dissected shark head, so if you are squeamish, please look away. But it is really beautiful if you're into anatomy. So this is what is going on inside of that shark head. That giant black sac, that is their nose. That is the area that is responsible for responding to chemical signals. And this is kind of how a shark smells. So water is moving passively. They can't sniff like you and I can. So water is going to move passively as they swim, and it's also not connected to their breathing. So whenever they breathe, they do not also smell like we do. It is a total passive movement. This is also really cool to study because this is a filter and a really high-powered sensor that doesn't ever clog. It constantly works, even though they're swimming through pretty mucky water sometimes. So when we study these guys and we think about how their nose works, that can really help us in developing some really nice high-powered filters or high-powered chemical sensors that can test water of different conditions. It doesn't have to be nice, pure water, and still work really nicely. So it has a lot of cool medical and engineering applications as well. So sharks swim through the water, and water will passively move into one of their nostrils. They have two nostrils on each side of their head. So it goes all the way in and then turns around and comes back. And if we look inside of that structure, this is what we see. So here is that in-current channel. So it is ending in that first nostril. So water is going in through that chamber. That's called the in-current chamber, so the way that water flows in. And then it turns around and comes back out the X-current chamber, which will terminate out here in this little X-current nostril right here. And if we look at that structure, what's inside that big black sac, we see something that is really impressive. And it is all of these individual lamellae. So each of those individual little tissues, they're kind of like dishes stacked in a dishwasher, right? They all kind of sit together. And each of those is where the smelling happens. And we can see these images really nicely and render them in 3D and do some nice modeling of how water flows through these structures. So that's just a nice view of them. So if we look at one of these lamellae and we turn it to the side, this is the structure that's actually smelling the water. So all within this lamellae, this little dish in a dishwasher, is all of these sensory organs that sit there and wait for chemicals to flow through the water. Those little sensors will grab the sensor and send that chemical signal and the electrical signal to their brain that says, hey, we're smelling something and it smells really good. So we kind of see a really giant diversity in shark noses, like I was saying before, and we're really confused as to why. Because, like I said, we don't see any differences in how well they smell. So why do they all look so different? And what we think is happening is we think that the way that water moves through these organs is really different depending on what the organ looks like. So to kind of take you to the opposite end of the shark spectrum, this is a pointy shark. So this is a Pacific spiny dogfish. And if we look at their nose, they kind of have a ball. So I call this like the tube to ball continuum, which is not super scientific, but it makes a lot of sense. So basically you have these really long organs and you have these really bally organs. And if you look at what is inside, that is a single individual lamellae. And we can pick each of these lamellae out and look at them individually and see how they're responding to how water is moving in these really different organs. So that is in blue, that is this one lamellae pulled out. So it's really cool new technology. We are basically pioneering how this is working in sharks, because a lot of times what you typically use is really dehydrating and it shrinks everything. But we have this new chemical formula that gives us these really nice, beautiful images. So again, that's kind of this tube to ball continuum. And that's what we're looking at now, is how does water flow differently in these really different organs. And just because these guys were on my little See and Learn promo thing, I just wanted to talk real quick about chimeras. They are ghost sharks or spook sharks or rabbit fish or rat fish. They have so many names. They're really creepy, and it's like spooky Halloween season, so I thought I'd talk about them. They only have one nostril on each side, and if we look inside their head, they look very weird as well because they have a starburst arrangement. They kind of look like a little daisy. So again, we just have really different sharks with really different noses, and you may be thinking this has gone really off topic. Now we're not at all on Ink, but we are on Ink, because what I'm really interested in about this is if water is flowing really differently through these really different organs, how is that going to respond to something sticky like ink? So ink will coat those structures. It'll really suck on those nasty chemicals. So ink is really this perfect defense mechanism that is going to really impact how water is flowing inside of those shark noses. So all goes back to ink. Really quick, the last thing that I want to kind of plug today is a really awesome organization, the nonprofit that I work with, which is Minorities in Shark Sciences. So a lot of you guys, if you guys have ever been interested or looked up shark science interns, you know, as you do always, internships are really expensive, and they are pay to play a lot of times. So you're paying a lot of money to maybe touch a shark once and dig a lot of holes or like fix plumbing for like 3 weeks. And that's really not fair to a lot of people who are trying to get involved, and it really does disproportionately affect minority women and minority genders, gender minorities of color. So we are an organization that provides these experiences free to our students. We are a hands-on organization. We get them in the field, we get them touching sharks, we get them taking samples and learning about shark science. We do professional development for them, and we do a lot of stuff to help diversify shark science, because if you've ever watched Shark Week, it's basically, they all look the same. Every expert they have on, there was actually a paper that came out that there has been more people named Mike on that show than there have been women in total in all the years of Shark Week. So we're actively trying to change that. So if you're interested in supporting us, we really appreciate that. But if you could just let somebody know if there is a gender minority of color in your life who really wants to study sharks but doesn't see herself represented and who is doing it right now, just let her know that we exist and there's people here for her, and we're here to help them. I really want to thank my funding organizations and everybody who sends me samples. Sharks are really hard to get a hold of. We never want to go out and kill a shark. So what we do a lot of times is we partner with people who are collecting shark samples. And sometimes, unfortunately, sharks do strand. Sharks do come up dead on the line. Sometimes fishermen are actively fishing sharks, and we want to work in partnership with them. We don't want to demonize them. So we have really awesome collaborators who, when they're done fishing their sharks, they will hand them to us, and we can scan them and get these beautiful images of their nose, like this one. This is one of the individual lamellae. So I really want to thank all of the fishermen that work with us, all of the nonprofits that work for us, and the National Science Foundation who funds this work. And with that, I will take any questions you guys have. Thank you so much. Cool. Thank you. Yeah.
Can we smell the ink?
Yes. So it actually smells. Oh yes. The question was, can we smell the ink? And yes, it actually is really interesting because ink, like squid ink specifically, it's a big part of culinary dishes in like Spain and in the Mediterranean. So squid ink pasta, paella, all these really awesome dishes. So we like that really bitter taste, but it does not react the same to them. So yeah. And if you come on my dive tomorrow, we will be taking ink. I brought some cuttlefish ink with me. So I'm basically loading like syringes with it tonight. And we're just going to be going up to fish and shooting them in the face with it and seeing how they respond. So hopefully we get those really nice diversions. And if we see a shark, I'll swim down and I'll shoot it in its face. Yeah.
I just wanted to ask you to talk talk a little bit more about your field project for tomorrow?
Yeah, so again, tomorrow it is at 1 p.m. That is my field project for tomorrow. We will be looking at these ink interactions in the wild. So we will be looking at this cuttlefish ink, and we'll be making them into these nice little vials, and then going out and seeing if we can replicate these lab experiments in the field, where we will look at how these fish in the wild are responding to cuttlefish ink being injected into their perimeters. We will also be looking for lettuce leaf sea slugs if we see them, because I do love sea slugs. So we'll be talking about lettuce leaf sea slugs and looking for them as well. Yes.
So you mentioned that the way that the water moves through the nose is this entirely passive but non-cloggable system.
Mm-hmm.
And because they are often swimming through really murky water, how?
Yeah, the question was, because I talked about how these are non-clogging systems and they are passive, so there's no water being forced through there. How is that working? And that is exactly what we do not know. So we don't know. Something that we think is going on, and I'm currently collaborating with somebody to look at the covering. Like they have, shark boogers. They have mucus just like we do. So we're looking at if the mucus itself is like special in coating the structures and making water pass really passively. I will say, just from like personal experience, this is not publishable data, but whenever I shot the cuttlefish ink, which was extra goopy, into my sharks, they hated it. And what they would do is they would swim around the tank really, really quickly afterwards. And what I think is they were trying to like get it out of their nose, because they got something really goopy in their nose and they were trying to irrigate it. So that is our best guess of how it's working, is they might just try to swim really fast and flush their nose really quickly. Thank you. Anybody else? Yes.
Was there like an inlet and an outlet in each nostril or each side of the nose?
Yes. So the question was, is there an inlet and an outlet in each nostril? And yeah, so they have two paired nostrils on either side. So it looks kind of like one or two nostrils depending on the shark. Sometimes it looks like one thing with like a little flap. Sometimes it's really, really separated. Like you can see two tubes. It's really different for each shark. And that's something else that we're doing, is quantifying like what their nostril looks like and what the inside looks like and how that affects the overall flow. Thank you so much. Awesome, thank you guys.
Thank you, Lauren. That was fantastic. I think everyone really enjoyed that. So if anybody wants to still sign up for the field project tomorrow, let us know. Otherwise, we will hope to see you on Thursday. And I don't know, what else? Where are we at? We good? Okay, so thank you very much, everyone, for coming.